Hello, my name is Kyle Quinn from the Department of Biomedical Engineering at the University of Arkansas. I'm a member of the Wound Healing Society Communications Committee, and our committee chair, Mitch Sanders, and I are pleased to host the monthly WHS Fireside Chat series to feature groundbreaking publications from wound repair and regeneration. This morning, we have a very special guest, my colleague in the field of biomedical optics, Kevin Eliceiri from the University of Wisconsin-Madison. We're going to talk about a topic near and dear to my heart, which is the development of imaging techniques to assess wound healing. Kevin's group has developed a novel method to image damaged collagen to better detect partial thickness and full thickness burns. Mitch can't join us for this session because he's taking a Friday off for his anniversary, but he sent me a few questions in advance to kick off this session. So Kevin, thanks for joining us.
My pleasure. Thanks for having me.
So in your study, you use collagen mimetic peptides, or CMPs, to identify damaged collagen. Could you tell us about this technique and its novelty?
Yeah, so this builds on the work of Ron Rains, who is a noted chemist who was at Wisconsin for many years and recently moved to MIT. And he was very interested in trying to build these compounds that would actually mimic the structure of collagen, this three triple helix structure, for lots of purposes, including the possibilities of using it for medicine and detection. And in our earlier collaborations, we were interested in using it for detecting collagen, such as our work in breast cancer, where we show that collagen was a prognostic marker. But more recently, in our collaboration with Angela Gibson, a burn surgeon, we were looking for ways to actually label the damaged collagen. As an optical person, I have done a lot of work with second harmonic generation, which is a powerful method of seeing the intact collagen, but it doesn't specifically label the collagen as it breaks down in a burn. And we wanted to see what happens when it starts coming apart and have a method that can actually look at that. And what's unique about these CMPs is because they mimic the structure of native collagen, they can come in and anneal on the broken strands. So it's a specific way of doing that. And you can actually attach your favorite fluorophore. So for this study, we did Psi5, but that could be changed, of course, too. So it's a way of fluorescently tagging damaged collagen. And so it's quite powerful because it complements other methods, such as second harmonic generation and other kind of antibody methods, but it gives unique information because it's damaged collagen specifically and not normal collagen.
Right. So you mentioned second harmonic generation, or SHG imaging, which you also performed in this study as a complementary method. And you referenced the work by Tanaka et al., who performed some in vivo second harmonic generation imaging on burns. Could you go into a little more detail comparing and contrasting SHG imaging and your CMP approach for assessing burns?
Yeah, so second generation is a favorite technique of mine, and that study of theirs was excellent. And we use it this study for the same reason, that when you can afford to, by that I mean you're looking at biopsies in a microscopy setting, second generation is actually a great complement to CMP because second generation, as you know, is very polarization dependent. And even though it is used to look at normal intact collagen, of course, if the burn or some other wounding effect affects the collagen, some of that signal will be extinguished, and you can use SSG in some form to certainly access normal collagen, but you also could use it to get some measure of how collagen is being damaged, but not specifically as CMP. On the other hand, we really need a way of looking at the intact normal collagen, so that's why second harmonic generation is great. The one thing I'll say, though, is that second harmonic generation, despite me loving if it happened, is unlikely to be a clinical method in terms of intact humans. It is, as its basis on a multiphoton microscope, and the fact that at most you can image a millimeter deep into tissue, and the fact that the form factor is microscopy based and uses lasers, and it's not endoscope based. Using it on an attacked human is impractical, and it's also quite costly and hard to manage. So a dye or a probe could be quite advantageous in the clinic because you could use that with a normal clinical wide field system instead of having to do second harmonic, right? But for this study, second generation was quite complementary, similar to what Tanaka did. So.
Right. So could you summarize the evidence that you have at this point demonstrating that CMPs are a promising biomarker for burn depth in the clinic?
So I think that part of the story is still to be determined, right? Because our paper here was really just proving that the CMP worked for damaged collagen in samples that were biopsied from humans, but it wasn't a full depth assessment. For that, we would really need to move to a different trial, and we would also need additional samples and also evaluate how we're actually going to image it, right? Because there are some challenges in terms of which modality and the bright wavelength and so on. As well, if depth is an issue in the clinic, there are some tricks that could be done to improve how the dye gets into the tissue. And we briefly discussed that in the future directions, but we might be able to think about delivery. So for this paper, think of it as proof of concept. This was really meant to validate this as a damaged collagen probe and not as a clinical probe. And that's why we didn't spend the time on getting it to work with a standard clinical microscope. That would be a future direction. So.
Right. And, so some of the wide field fluorescence guided procedures that have gained some traction in the clinic generally lack any depth resolution. So are you thinking about other techniques or iterations of those current techniques to provide some additional depth resolution, or do you think that's not necessary? You can really just pull that from the intensity of the fluorescence?
I think that remains to be determined. In other parts of my lab, also full disclosure, I have a spinoff company called Onloom, which makes instrumentation for the surgical clinic. So they may do some things in this area. They currently have an FDA-approved device for looking at extrinsic things like ICG and other kinds of approved fluorophores. So that system could be adapted for this pretty easily. So I think that there are some things to be done there. But when you think about depth, even with some optical tricks and computational tricks, we may still need to look at how the probe itself is delivered and think of ways of getting that deeper in. But certainly, I think that as we do additional trials, that will be a focus.
All right, thanks. So Mitch would like to know whether this could be commercially feasible as a burn depth biomarker, and is this technology patented and available for licensing?
So that would have to defer to my colleague Ron Rains, because that is the actual CMP is his technology, but he has patented forms of this, and he is involved in some commercial ventures for other applications. So as we prove this in papers and there's interest, I'm sure that Ron will be interested in pursuing that. And if there are folks that read this paper interested in that, definitely should contact us about that. But certainly there's potential there because it is a novel probe.
So we kind of touched on this, but what do you see as the next steps for advancing this technology as a wound depth biomarker? Is the focus solely on clinical translation, or do you envision this as also a tool for wound repair laboratories developing products?
Well, certainly the applications in research are quite deep, and also the road to entry is much simpler, right? Because you would have to go through clinical approval. So yeah, I think for people that are very interested in the role of the matrix in disease of any kind of wound healing, not just burns, the potential of using this as a probe for damage is very interesting, right? Like I have a general interest in all types of wound healing. I use wound healing as the most generous term. So to me, wound, it can be anything from a cancer to a cut to an abrasion to an implantable electrode to a burn. So we too often don't look at how the role of collagen plays, but particularly what the role of damaged collagen plays. So this could be a useful counterpart to what's already out there. As well, there could be other possibilities of this, using it for drug delivery and targeting. We and others have shown how important collagen matrices are, and Ron's work has really shown in his other papers how he can really mimic the collagen matrix. So you could imagine all sorts of agents that could be used with this, either for repair or for targeting, for drug delivery. So I'm quite interested in that from a research point of view. Clinically, we have this tremendous collaborator who's one of the other senior authors, along with Ron and I, Angela Gibson, who of course would love to see this in her papers. And there's another paper that also is in WR that's coming out soon, or maybe already is out with her as senior author, where we're trying to create better models of burns in rodent models. And this could be very useful there because the more we go in vivo, the better, right? So I would think that one of the next steps with the CMP work would be to go into in vivo in the rodent, just because of the full control and really wanting to validate everything before we move in vivo in a human.
Right. So having visited your lab a few years ago, I know firsthand you have a lot of fun optical rigs and analysis tools that you're using really for a range of biomedical applications. Do you have any other novel techniques that you'd like to talk about that you're developing for wound healing?
Yeah, so there'd be a few things. I think I saw, one of the interests that one of the questions that was on there was, my conflicts of interest with this company OnLoom. We developed a technology in my lab first that got patented now as a spinoff company called transient lighting, which is the idea that we can modulate the room light to such a high frequency, 300 hertz, that your eye thinks the lights are always on, but they're actually blinking, and we can set the detector. And for folks in the wound healing field that do imaging with fluorescence, either in clinical models or animal models, where they want to look at weak fluorescence in an attacked model, this is quite attractive because then you can have a BOOMR microscope, either a clinical microscope or animal scope, that could look at very weak fluorescence with all the lights on. And particularly for human surgery, that's a really big deal to be able to do that. On the research side, similar to you, I'm very interested in metabolism. And one of the things that we're looking at in other studies is what role does metabolism play in the wound? And I think that when you think about the cellular microenvironment, which sums up all my research interests, the metabolic and the ECM are really interesting components of the wound, and we want to characterize that not only in burns but other kinds of wounds as well. And I'm very interested in cataloging these changes. But I think it'd be fair to say that most of my interests have to do with how can we characterize the cellular microenvironment in different kinds of wounds and see what is preserved or what is different in a wound, right?
Yep, I totally agree with you in terms of the interest in metabolism, and there's definitely a lot of applications there. Is there anything else that you'd like to add about this current study?
I think it's an important first step. I think it points out that there is a need for better probes. Our field is starving for probe development. I think that we have tremendous computational people and tremendous optical people, but there does need to be better probes and more probe development. So if anybody out there is interested in developing probes, I encourage them to think about these kind of problems because there needs to be better agents. And as much as I love autofluorescence, which is a big part of my career, specific extrinsic fluorescent tags are really important, and they really allow us to get more mechanistic and also make clinical relevance clear. So.
All right, well, Kevin, thank you for joining us this morning, and I'd encourage
Okay.
everyone to take a look at his group study currently in wound repair and regeneration.
Thank you so much for your time.